love the part that aligns iGEM with Ginkgo in the development. And as Austin mentioned, the cost of doing synthetic biology is still not cheap enough. But in the past decades, the advancement of DNA writing, DNA synthesis, has significantly increased our ability to engineer biological systems. And thanks to IDT, iGEM teams have been receiving free DNA synthesis and other tech support to accelerate your project progress. This is the 10th year that IDT collaborates with iGEM. Let's welcome Sandy Ottensmann, VP of Gene Writing and Editing at Integrated DNA Technologies, onto the main stage. Welcome, Sandy.
Thank you so much. It's wonderful to be here with everyone at iGEM and be here representing Integrated DNA Technologies with all of my colleagues who are here. You know, as we said, IDT has been a sponsor of iGEM for 10 years, and it has been amazing for us to be a part of this and see all of the great work you have all done. So I'm here to actually celebrate 3 different things today. First is to congratulate iGEM on 20 years. It is amazing the technologies, the products, and the companies, like we just saw from Ginkgo, that have come out of this competition. And from IDT's perspective, we are celebrating 10 years as a sponsor here. I'm going to talk about it in a minute, but IDT is a company started by scientific researchers like all of you, for scientists. And we have been evolving the science of genomics and oligo DNA synthesis to support all of you. So as was just mentioned, we have, during these 10 years, provided to all iGEMers 20 KB of free gene fragments, or equivalent of our CRISPR products or our DNA oligos. Can I get a raise of hands or a shout if you used IDT projects in your project? Awesome. We are so excited that we can help you support in your research, and we were able to accelerate that. I got to talk to a lot of different teams since I've been here this week about how IDT was able to get you products next day, really help your research, and how much this was able to drive what you all did going forward. So we're really glad we've been able to support all of you through that work. But I also want to celebrate one more anniversary that we are celebrating here at IDT, which it's IDT's 35th anniversary this year. Now, that might seem a little surprising, actually, if you think of how fast and innovative genomics and synthetic biology is, that IDT was born in the 80s. We're a millennial. And if you think about that, like, that's a long time. And what was even synthetic biology and DNA like back in the 80s? And I want to tell you a story that was relayed to me by our Chief Scientific Officer, Mark Belke, who was hired by IDT's founder, Dr. Walder, very early on to run R&D. And Dr. Walder told him that he had a vision. Dr. Walder was in the University of Iowa, a researcher, and he had a vision that DNA was going to revolutionize modern biology, the ability to synthesize DNA. And he told Mark, he's like, we need to start a company to do this, to enable researchers, because synthetic oligonucleotides are going to revolutionize modern biology, revolutionize therapeutics, and eventually medical diagnostics as well. Now, for all of you here, you're probably like, well, duh. Like, that seems obvious at this point. But remember, this is back in the 80s. This is more than a decade before the Human Genome Project finished. This was way ahead of our time. But Dr. Walder realized, OK, we need to start a company to make oligonucleotides because we can't make them fast enough in our lab. So he came up with the first plan for IDT. And do you know how many oligos he said IDT would make in a year? 10. How many of you use more than 10 oligos in your project? Probably a lot of you, right? Probably hundreds, thousands maybe. So how do we go from 10 to where we are now? So IDT started with 10 oligos, then went to 100 oligos a day, 1,000 oligos a day. Then we started offering RNA. Then we found out our customers, and we've always really collaborated with researchers and asked you guys, what do you want? Okay, you want PCR products. We will come up with PCR. We'll do products for next-generation sequencing. And then our customers came to us and said, we want to start diagnostics with these. So we're like, OK, we'll get the 1345 certification, which is an ISO certification required in order to make these products for diagnostics. Then we invented the G block. So IDT invented the G block because we pushed DNA synthesis longer and first. And then when CRISPR came on the scene, we started offering products for that too because everyone was clamoring for it. And throughout this, we scaled our synthesis. So here's a question, audience participation. How many oligos do you think IDT makes in a day? Any ideas? Shout out. Thousands? Millions? Not quite millions. 100,000 oligos per day. It's actually 130,000 oligos per day that we make in our sites in the Americas, in Europe, and Asia Pacific. That's 5 bacterial genomes a day of DNA. That's 135 million bases per month, or 1.4K of gene fragments per day. That's a lot of DNA. And if it wasn't for us being able to help push this over the last 35 years to where we can actually make this much DNA, a lot of the projects that can happen now that you're all working on, a lot of the research wouldn't be able to happen. So now we have, as we've scaled that, had to figure out how to make that a lot more sustainable. Because it's a huge process, it's a huge manufacturing process as well. So this is a diagram of our main facility, which is in Coralville, Iowa, which is still where our headquarters is. As I said, we're a spin out of the University of Iowa. And just the steps we've taken to make our building more energy efficient. We've really also pioneered how do we recycle chemicals and all the things that we use in our processes, because we know that unless we can make this process more sustainable and make it work better for the planet, we're not going to be able to do all of the amazing things that synthetic biology can do. So I want to step back for a minute and say why I'm so excited about what we do at IDT. And I started on the bench. I was a scientist, like all of you, in school, in my first jobs, and I worked on a lot of different techniques. So I did a lot of PCR. I did a lot of HPLC to purify and analyze my products, and I used a lot of equipment. I used a lot of centrifuges and plate readers and mass specs and all of that. And what I've gotten a chance to do in my career is now work for a number of companies, including IDT, Phenomenex, and Beckman Coulter, who make a lot of the products that I was able to use. And I've actually gotten to work with product and actually make products better that all of you use every day. This is through a company called Danaher, who actually owns all of us. It's a leading science and technology firm, and our objective for Danaher is to innovate at the speed of life to help drive medicine and the human condition forward. And that's where I kind of want to go with the next part of my talk, which is where is iGEM going to go in the next 20 years, and what does IDT think is going to happen in the next 35 of genomic advances in synthetic biology? So I mentioned that IDT was founded to make oligos and to make a lot of them, and we've done that, but that was step one. Step two of that vision was to innovate for medical diagnostics and therapeutics. And we realized we had to change a little bit of who we are to become not just an oligo company, and that started with our mission and vision, which is to accelerate the pace of genomics and enable researchers to rapidly move from the lab to life-changing advances. So we now want to partner with scientists and researchers like you and academia and industry on how do we help partner and push through these innovations for human health. So what we've done is we don't just make oligos. And I think I've had a couple people come up to me today and they say, you make oligos, right? And we said, yes, we make oligos, but we also are working on solutions and how we drive it forward. We have more than 100 R&D scientists, not focused on oligosynthesis, but focused on how we're working on applications and working with customers to come up with these next solutions. Obviously, we have PCR products. I'm sure many of you use those in your research. We also have a lot of projects for NGS, for next-generation sequencing. In fact, we acquired a company called Archer last year that is driving clinical oncology research, helping us really drive diagnostics in oncology. As I mentioned, we have a huge presence in synthetic biology. I'm sure many of you, you've used G-blocks or some of our products there. And then again, CRISPR technology as well is something we've really pioneered it on the research side to make as easy as possible for people to use. And these last two are really where I want to focus for a second, because I could talk about so many different things, and you can look around this entire room and see all the different applications, whether or not it's food science and agriculture and energy and the environment and sustainability and biomanufacturing and diagnostics. But I want to talk about therapeutics, because I am really excited about where we can help push the science forward to really, truly cure diseases using genomic medicine. So when we talk about genomic medicine, we're talking about using the human genome, whether or not it's in general or for an individual patient for personalized medicine, to make actual therapies that can cure disease. Not treat disease, but actually cure disease. And we can do this through gene and cell editing. So we can do direct gene editing, things like antisense oligonucleotides, or ASOs, and RNAi, which help knock down a gene. These have been around for a while, and there are many on the market. But then what we can do now with CRISPR is we can actually edit and knock out a gene completely, or combine it with viral gene replacement or mRNA gene replacement to actually add new functionality. Now we can do this themselves or combine it with cell therapies. Now CAR T, stem cell therapy, you've heard of those. They've been around for a while. But now that we have CRISPR and the ability to use CRISPR and synthetic biology to really engineer these more precisely and more accurately, this area has exploded in the last number of years. And I'll talk about CAR T in a minute, but for stem cell therapies, the really exciting one that I'm sure many of you have heard about is around sickle cell disease. So what you can do is you can actually take out the bone marrow stem cells, engineer them to restore the fetal hemoglobin, and then put it back in the patient, and you can actually cure sickle cell. That's never been possible before. We can treat sickle cell, but we can't cure it. And Vertex Pharmaceuticals and CRISPR Therapeutics are about to have any day now approved the first CRISPR treatment for therapeutics. They actually presented at the FDA earlier this week and got some really great feedback. We are so excited for them, and we're so excited for all the companies who have an incredible amount of clinical trials that are going through right now that are driving in this area, and we're only going to see more as CRISPR opens up the ability for new therapeutics. So I know many of you used CRISPR in your research. For those who didn't or need a little bit of a primer, this is how we use CRISPR and synthetic biology to edit genes. So CRISPR, we make, and IDT makes this. It's the guide RNA. It's normally around 100 mer that matches to the DNA and tells an enzyme like normally Cas9, but it can be a few others, exactly where to cut, and makes a double-stranded break. Now, you can either let those recombine and non-homologous gene editing, and that it works. It just it knocks out the gene, but it's not as precise. What's really great is we combine it with a synthetic biology, an actual gene, and then with homology-directed repair, you can modify or you can build new gene functions. Now, what we can do with this now is we can use this for medicine. And this is one application that I want to just, as an example, that's being used in clinical trials right now is for CAR T cell therapy. And the way CAR T therapy works is you take T cells out of a patient, and then you engineer them to make them better at finding and attacking the antigens to attack cancer. And so if you look at the upper right, what has traditionally been done is using a viral vector to incorporate the gene, but that has some risks because the viral DNA can actually get in to the cell. And so the industry is sort of moving a little away from that. Doctors are moving away to looking at synthetic biology and CRISPR ways of incorporating the CAR receptor, that's what you really need to make it that CAR T cell to better attack the cancer. So what we can do is the CAR is actually a 2 kb. We can make that into a gene donor block for HDR donor, and then we can use CRISPR. And CRISPR, in the bottom right corner, is you can do two things simultaneously with the CRISPR. One is you can incorporate that gene for the CAR while simultaneously actually knocking out receptors and genes in 3 different receptors. That knockout is important because it actually makes the T cell work better. It reduces graft-host disease and T cell exhaustion. So now what we've done is we've knocked out some things that make the T cells last longer. Then we've added this new CAR receptor that's needed to make it the CAR T, and you can put it back in the patient. If we do that for one single patient, it's an autologous therapy, but there's also an opportunity to get this from healthy donors, and then an allogenic therapy actually use healthy donors, engineer the cell, and go to many patients. This is just stuff that wasn't even thought about years ago, right? We had T cells, but the ability to do this is opening up a lot of medicine. IDT is collaborating with partners who are driving this into the clinic right now, a lot of clinical trials, and you're going to see more of these get approved in the next couple of years, especially as we see that first CRISPR therapeutic get approved. So I'm really excited about this, and I'm excited about what we're doing. IDT, along with its sister company at Danaher Aldevron, actually supported, either in research or up through therapeutic development, 75% of companies that received FDA approvals for cell and gene therapy last year. And we are really excited to continue that work. We have been a leader in CRISPR, actually, and I do want to pause and take a second for that. You know, our researchers, the day in 2012 that first paper was published on CRISPR, our scientists were already trying to figure out how to do this so we could enable researchers. Probably most of you back in 2012 weren't yet doing science, but we knew that this was going to be huge, and we had people working to be able to offer guide RNAs, and eventually then our Cas9 and Cas12 products for CRISPR to make it as easy as possible for people to do research. We came to market with a lot of actually first-to-market products on this as well, electroporation HDR enhancers, and then also we use NGS to do off-target analysis to see if the CRISPR is editing the right thing. All things that we've done, but we were missing something. And for us, we were helping researchers, and the pace of CRISPR has been so fast to get to the clinic. As I said, we're going from like 10 years from discovery to first approved therapeutic. That's lightning fast in therapeutics and with approved drugs. And we did not have the ability to support our customers as they moved, our researchers, as they moved into the clinic. They were like, okay, great, we've been using your products in research. We now want to do clinical trials. We had to say, no, we can't support you until now. So we're really excited that we actually just launched 2 weeks ago in Iowa, where our headquarters is, a new therapeutic oligonucleotide facility. We lovingly call it TOM. It's our new facility where we're making CRISPR for therapeutics, for clinical trials, and for eventual therapeutics, so we can actually partner with a lot of the researchers we work with to help them get into the clinical trials. And this is, when I go back to the 35 years of IDT, now gets us fully to Dr. Walder's original vision of revolutionizing therapeutics. And we've gotten there with this new facility and this new capability. So for all of you who are doing research in anything for therapeutics this week, know that we can now support you if you're also trying to get and take that into the clinic. So this isn't it for any of the research we're doing. One of the exciting things for IDT has been that every time we have pushed the boundaries, making the first G block, making our first CRISPR products, people come to us and said, that's great, but can you make it longer? Or can you add this modification? Or can you do this instead? And we joke that literally every time we launch a product, we get that, and we absolutely love it. So I'm hoping that you all find us at some point, find me, find someone else from IDT at our booth this week, and tell us, we've loved using your products, but if only you could do this. Please tell us. That is what has driven us for 35 years, is how do we push the boundaries of how to make the products that will drive all of the work you guys are doing. Because we are truly in the golden age of biology, and you guys are all the ones in the next 20 years who are going to be driving this. And we have every intention to be here to support you, and we can only do that if you let us know how we can best help all of you. So with that, I just want to thank you all for being here. I want to congratulate all of you and all of your teams for all the amazing work you have done and getting here today. Best of luck to all of you in the competition, and have a wonderful rest of your week, and thanks for having me here. Thank you very much.
Indeed, like we expect more and more iGEMers join the force, solving the challenges we are facing today, and also build the future that we all want to live in, designed by biology. And that's all for the main stage today. The Plaza sessions will resume soon, so take a short break and join the 2023 teams and our amazing expert speakers. Another thing to highlight is that the second iGEM Responsibility Conference in collaboration with OECD will continue at 1:30 p.m. in workshop room 1. So don't miss the opportunity to actively engage and deliberate in the discussion about what kind of future you want to build with synthetic biology and how we shall work with that. Thanks again for joining us at the main stage today, and see you all here tomorrow. Thank you.